Hello, thank you, Stephen. So welcome back, everybody, for session 2 on day 1. We have our keynote speaker up first. So Ben Ineichen, who is joining us from the University of Zurich. So Ben's okay, we're at 3:10. Stephen, are you happy for me to go ahead?
Yes, absolutely. Thanks, Claire.
Thank you. So welcome back, everybody, to session 2 on day 1. I just wanted to do a little reminder for those of you that might just be joining us, if you're joining us live, that you have a little question mark icon that you can click on to add your questions as we go through the talks for this session. So we have 3 talks in this session, all on really interesting topics. So we'll get started straight away with the first talk. So the first talk in this session is a keynote talk from Professor Benjamin Ineichen. Ben is joining us from the University of Bern in Switzerland, and his talk today is about widespread post-approval animal studies and the implications this has for drug research. Thank you.
Since doing my PhD in animal research, I've been wondering how efficient is the drug development process from animal testing to clinical testing to regulatory approval. And we could finally look at this last year, where we did a systematic review on a list of around 370 therapies for more than 50 unique diseases. For each of these therapies, we checked how far along this pipeline does the therapy move from animal testing to clinical trials to regulatory approval. Surprisingly, every second therapy tested in animals actually made it to a clinical trial. However, only 5% made it to regulatory approval to be used in clinical patients. So that means only 1 out of 20 therapies reaches the approval stage for human use. And this was covered widely by media, this finding that only 5% of therapies tested in animals obtain regulatory approval for humans. And I'm not showing this to brag, but I'm showing you this to tell you that you should not always believe what you see in the media. Because there was this Italian news outlet which translates to only 20% of novel drugs obtain regulatory approval for human use. But it's 1 out of 20, which is 5% and not 5%. And with this, hello, I'm Benjamin. I'm a professor in medical data science at the University of Bern in Switzerland. And I'm excited to talk to you about this topic, which is very close to my heart, which is post-approval animal studies in drug research. Very briefly about our research group, we are the STRIDE Lab. STRIDE used to be an acronym for something, but we don't remember what it stood for, but we liked it, so we kept the name. And the focus of our research is to understand the drug development process better. Which factors drive successful drug development and how can we make this more efficient? So I've shown you that drug development is a very inefficient process. 5% of therapies make it. I mean, 5% is still a lot actually, if you look at it closely, but still 5% is a relatively low number. And so let's assume there's 2 drugs. There's a blue pill and there's a red pill. Both of them have been tested in clinical trials in humans, and the blue drug obtained regulatory approval and the red drug did not obtain regulatory approval. It dropped out of the testing because of safety or efficacy concerns. So drug development is obviously a very innovative process, and there will be failures, no matter how you do it. This is just an inherent feature. However, there might also be factors driving successful drug development. For example, maybe certain outcome measures in animal studies or in human studies are better suited to predict how well this drug will actually perform in a clinical setting, or maybe certain trial designs or the trial rigor, like randomization or blinding of the clinical trial or animal study, matters whether a drug will obtain regulatory approval. We don't know, but we can potentially figure this out, and this is the focus of our group. And we use one of our main tools is the systematic review. So if you're not familiar with this method, in contrast to a classical review article, a systematic review uses explicit methods to address a scientific question, and it thus mitigates common biases associated with traditional narrative literature review. For example, you screen through the literature, you do comprehensive literature searches, and you look at each individual reference and checking, is it relevant to answer my research question or not? And if you're interested, we have recently published this primer, and I'm happy to share a copy with you. And we have used this systematic review methodology for a cool study, I think. This study has been conceived mostly by 3 medical master students in my group, Ingrid, Pia, and Wolfgang. And we looked at drug development for multiple sclerosis. Multiple sclerosis is a very interesting disease to look at for this aspect because it has quite some drugs which are used in clinical settings, so which have obtained regulatory approval. But of course, it also has drugs which have been tested in clinical trials and animal studies, but which failed at one point. And the idea was that we look at these drugs and go back to the animal studies and check, could we have predicted that one drug obtains approval and the other drug do not obtain approval. And we have done a systematic review, so I will spare you the details, the technical details, but we have looked at around 500 animal studies in detail. The first interesting thing was that these animal studies used a very narrow set of experimental parameters. More than 80% of the animal studies used mice to test these drugs. And there was, which is quite surprising, a very strong female sex bias. Usually you have a male sex bias in experimental research, but in this case it was a female sex bias. So more than 80% of the animal studies exclusively used female animals to test these drugs. Second, we aim to understand whether effect sizes in animals predicted the later drug approval status. So the hypothesis was that maybe stronger drugs in animals were more likely to gain regulatory approval later. And we looked at EAE scores. EAE is a common animal model for multiple sclerosis, which is measured on a score from 0 to 5. The EAE score, the higher the score, the more functionally impaired a mouse or a rat is. And you can see here on the right side in the graph, in blue the approved drugs, the later approved drugs, in red the later failed drugs. You see in black the effect sizes in these animal studies was exactly the same between the approved and failed drugs. So no association to clinical value. Third, we hypothesized that maybe if you test a drug in more diverse experimental settings, so for example, if you test it in more than one MS animal model or in both sexes or in more than one species, maybe this is associated with a successful drug approval. So we looked at approved DMTs. DMT is a drug, a disease-modifying therapy for MS in that case, and failed disease-modifying therapies. And we didn't see a difference again here. So apparently testing in a broader, more variable setting was not associated with drug approval, to our surprise. And fourth, the most striking finding is this. So we looked at the timing of these animal studies. And if you focus on the left part of this graph, you see the approved drugs on the y-axis. And on the x-axis, you see the time scale. Each orange dot indicates when this respective drug gained the official FDA approval, which means that this drug can then be used to treat human patients. And each gray dot is one published animal study. And you can already guess it from these data. The majority of animal studies are published after the drug has gained regulatory approval, is used in clinical settings. And actually, when you quantify that, it's more than 90% of the animal studies have been published after first in human trial or after regulatory approval. And you've then, of course, wondered, is this a phenomenon exclusive to multiple sclerosis research, or is this a systemic phenomenon that there is lots of post-approval animal studies? And Pia was also involved in the other study. She's a medical master student. She has painstakingly looked at many thousand articles, and I will show you this now. So in this study, we have expanded the scope. So we looked at all the FDA-approved drugs which obtained approval between 2011 and 2014. And we excluded non-drug treatment and a couple of other things. Then we included primary studies testing these drugs in animals of any sex or species in an animal model aiming to resemble the human indication. So for example, we included all studies which tested a drug approved for diabetes in a diabetes animal model. So not just a certain drug in any animal testing, but for this particular indication, so a model for the particular human indication. And we excluded in vitro studies and a couple of other things. And again, this is a systematic review, so we search different databases, in our case PubMed and Embase, to be really comprehensive with what we look at. And we have specified all in the protocol, including what data we are going to extract. So first we extracted data related to study characteristics, including the publication year, of course, of this study, which animal species and sex have been used by the studies, and the study country. Second, very importantly, we also checked in each paper, how did the authors argue why did they conduct the study? I'm presenting all the data here, but just to give you some examples. The first one is mechanism. So for example, the authors claimed that they want to understand the drug mechanism better, and that's why they conducted the animal study. Or reason 6, efficacy. The authors claimed that they want to check whether this drug shows efficacy in animals. And third, we also extracted effect sizes from these animal studies. So how strong were these effect sizes in the animals? So we included 62 drugs initially. We excluded quite some of them because they didn't meet our inclusion criteria, and we ended up with 36 included drugs in total. And I'm listing them here. Obviously, the details are not important. You can pause if you're interested. I'm showing this to show you there was a broad range of different kind of molecules. There were small molecules, there were biologics, and there was also a broad range of human indications, from cancer to immunological diseases to neurological diseases. So a very broad panel of indications. Here's the flow chart for the study inclusion. So our database search yielded more than 20,000 individual studies, and Pia went through all of them, or through most of them. And we ended up including around 1,600 studies, which tested these drugs in an animal model for the relevant indication. And here's again the data in the graph you are already familiar with. So on the y-axis, so on the vertical axis, you see all these 36 drugs, and on the horizontal axis, you see the year. And again, orange is the approval year, and gray is one individual published animal study. And Again, 84% of animal studies are published after regulatory approval. And you might say now, well, that's publication. That's not we don't have information about when has this study actually been conducted, which is different, of course. We don't know that, I can tell you. However, we have somewhat looked at that. Even if you build in a safety margin of 3 years, so we have looked at the literature, there is a couple of studies showing that it takes roughly 3 years from writing a study protocol, submitting documents to the ethical committee to gain approval for the animal study, and then conducting the animal study, writing up the findings, submitting the manuscript, and the paper being published. That takes roughly 3 years on average. And so even if we have this safety margin of 3 years, it's more than 2/3 of studies, of animal studies, still being published and likely conducted after the drug has gained regulatory approval. And by the way, we looked at this, most of these studies were from the academic setting. And academia usually doesn't have a vested interest in intellectual property, so there is not really a reason why researchers would withhold their findings until a drug gains approval. That might be the case for industry, but academia, usually PhD projects and all these things, they need to get published as soon as possible. So we looked at the characteristics of these post-approval studies, and then in the bottom panel in red, you see the post-approval studies. Most commonly, mice were used for these studies, and in this case now there is a strong male sex bias, so much more male animals were used for this drug testing, and the most prolific countries were the US, China, and Germany. And now, of course, we also looked at the reasons. Why did the authors conduct these studies? So on the right again are the post-approval studies in red, and you can see that efficacy was by far the most reason mentioned by the authors of these studies. So even though these drugs have gained approval to be used in the clinical setting, the authors aimed to test for efficacy in the animal experiments. Other commonly mentioned reasons were benchmarking or focus drug, which both of these classes say that the drug has been used as a comparator to another drug. And understanding a mechanism better was also another commonly mentioned reason. Importantly, 22% of post-approval studies exclusively said that they're for efficacy reasons. So some of these reasons are overlapping, and an author might state more than one reason, but 22%, so every fifth animal study was for efficacy reasons only. And finally, we also looked at, well, if efficacy is so commonly used as a study rationale, does efficacy in animal studies actually predict or is associated with the actual drug value in humans? We looked at this here, and no. Efficacy or the effect size of drugs in animals was not associated with the actual effect size or the value in the clinical setting. So let me summarize. The majority of animal studies are published and likely conducted post-approval. Efficacy, benchmarking, and better understanding a drug's mechanism is a commonly mentioned rationale in the study. Around every fifth of post-approval studies are exclusively conducted due to efficacy reasons, and effect sizes of animal studies were not associated with clinical drug value. You have to be aware of certain limitations of our study. First, we use the publication dates, so we don't have formal data when these animal studies have been conducted. However, this has been addressed. I showed you the data when considering a 3-year time gap. And second, the studies were mostly by academia. And so I think it's likely that still the most of these animal studies have been also conducted post-approval. Second, harking. You might have heard of this. Harking stands for hypothesizing after results are known. And this is a questionable research practice where basically authors reframe why a study has been conducted in a paper in contrast to how they initially, or what the initial goal of a study was. And third, publication bias, that's the Achilles heel of systematic review. So not all things are getting published because, negative findings and other reasons, but we cannot control for that. So what does that all mean? As a main take-home message from my talk is that post-approval animal studies are very common. But what are the reasons for these? So one reason is, I think, upon approval, the drugs become available to researchers. So this is called reverse translation, which means that human research informs animal research. And I think this is a good thing because this way we can make animal research more relevant for human research also. I also think that there is a disconnect between animal and human research. So some of these studies have been conducted, for example, to test for efficacy in the animals, even though the drug has already proven its efficacy in humans. And in this scenario, the harm-benefit ratio in the animal studies does not justify the conductance of these animal studies potentially. And this can pose research waste. We call to action to first foster the collaboration between animal and human researchers. Second, to train animal research better on the real-world limitations of translation. And third, better understand the drivers for successful drug approval. And this is what we are actually focusing in our research. So you might know there's a very steep increase in the number of publications related to animal research or any other field actually. And this makes it actually practically impossible for researchers to keep abreast with these data. We need methods to still keep up to date with these data. And we actually use these models to better understand this process. So all of you know at this point ChatGPT. So we use the models behind ChatGPT, open source versions of course, to mine millions of publications related to drug testing in animals and humans also. And we feed these data into a central data warehouse, which we later interrogate to ask which factors drive the successful approval of drugs. And with this, I would like to thank my collaborators, my team who has done a tremendous job in all this. And of course, the funding from the UFAW, which was instrumental for conducting this research, and other funding, of course, as well. And all of you for your attention. I'm happy to hear your thoughts on that. Thank you.
Thank you very much for that talk, Ben. I'll just give you a second to get your camera and mic on. Hi, Ben. Are you okay to unmute yourself?
Hello. Yes. Hello, everyone.
Hello. Thank you. So quite a lot of questions coming through on the question chat here. So I'm just going to sift my way through and see how many we can get through in this time. So picking up on first question from Lorna, she says, excellent work, very well presented, and thank you. She's asked, were all of the studies for each species or drugs used for exactly the same clinical indication? I'm not sure if you got that detail on the systematic review that you were doing, but were all the studies for each species and drug used for the same clinical indication?
Yes, thanks. Thank you. Very relevant question. Yes, we did. This is, I think, very important. For example, we only included, for a drug approved for diabetes, we only included diabetes animal models, for example, or a drug approved for brain cancer, a certain type of brain cancer, we only included animal models of that certain type of brain cancer. I think that's a very good question, and it's good that I can clarify it now again.
Thank you for that. And a question from Baz Rodenberg. So I think this was relating to the initial systematic review that you mentioned. This question came in quite early in your talk. So he asked, do you know what the reason is for the female bias in that initial systematic review? I know your later one had the male bias. He's asked, is this to compensate for older studies, perhaps where there was more of a male bias in the sample? And would it be better to have studies with a more balanced sex representation?
Thanks. Yeah, that's an interesting question. So there is actually in clinical research, I'm a medical doctor originally. In clinical research, there is also a huge male sex bias. It's like a very male-dominated field from the patients which are included. And in experimental research as well. The reason why there is female animals in MS research, there is this female sex bias. I think the reason is that I've worked many years with this EAE model, and it's much easier to induce in female animals different immune systems, and it usually just works better. It's easier to establish in a lab in female animals. That's likely the reason for this. And of course, I would argue, the second part of the question, I would argue that we would, I think, need a better representation of the sexes in both clinical and animal research. Although I must say, we don't have empirical data showing that this would actually improve the relevance of animal research to humans. So we don't have empirical evidence, but at least it would seem plausible that we should have that.
Thank you, Ben. I've got a question now from Hugh, actually, and this addresses what you're talking about in terms of sort of implications and how we can use this data going forward. So Hugh's asked, would pre-registration of post-approval studies help with many of the issues experienced here, especially if pre-registrations were peer-reviewed before going ahead with the animal studies? I'm not sure what your thoughts are on that proposal.
Yeah, pre-registration is a favorite topic of mine. I just attended a workshop last week, if we should make pre-registration of animal studies mandatory in Switzerland. I think, again, actually the data, we don't have so much empirical evidence showing that pre-registration would improve the scientific quality. Some people say it just fights the symptom, but not the root cause of these problems. But I'm a strong proponent of pre-registration. I think if other potentially research peers could evaluate these post-approval studies in advance, this might help to reduce some of these animal studies, which some of them I think are unnecessary and are not justified from an ethical harm-benefit ratio.
Thank you. And Susan has asked, are there some studies, some of the post-approval studies that are perhaps related to novel application versus original labelled use? And did you look at that data in that review? Because Susan's mentioned that that's actually common in veterinary drug approval.
Could I kindly ask you to rephrase the question?
So for the post-approval studies, were any of those related to using the drug in a novel way rather than for what they originally had the FDA approval for?
Okay, yeah, that's a good question. So I could interpret in a different way in different ways. But what we saw is actually one of the 10 categories was also regimen, for example. So regimen was testing a novel drug regimen, for example, giving it intravenously instead of subcutaneously, or giving lower doses over a longer time instead of higher doses over a shorter time. That was another category which was relatively rare in our study, and this would be one different way. Would that answer the question?
I think possibly a question.
Yeah.
Yes, I think so. Thank you. So I think we've got time for one more quick question here then, Ben. So Elizabeth has asked, back to the methods, I guess, of the review. So why that time frame for the study? So 2011 to 2014, why specifically that time rather than a broader time frame for the review?
Yes, so this was a convenience sample. The idea was that until 2014, we started a study in 2024 last year, so that we had 10 years of post-approval time to basically have some time passed to post-approval studies being conducted and published. And as I've already shown, it was already with these 3 years, 2011 to 2014, we had more than 20,000 studies, 36 drugs. So it just, if we had more, I think it just becomes at one point unfeasible. So convenient sample.
Okay, thank you. So potentially some more work to be done to look for more broad samples in the future. Fantastic. Thank you very much, Ben. So that ends Ben's talk and questions. We'll move on to our next talk for this session. So next we have Catherine Arpin's talk. Thank you, Ben. So Catherine's talk, Catherine is from McGill University in Quebec, Canada, and Catherine's talk is about the study of behavioural needs of adult dairy cattle across common intensive housing systems. Head over for Catherine's talk.
Hello,
thank you.
Everyone, and thank you for coming to my talk. My name is Catherine Arpin, and I'm a master's student at McGill University in Canada. And today I will be presenting my scoping literature review on the behavioural needs of dairy cows across common housing systems. So before jumping in, I thought I would make a little example to introduce the general concept of behavioural needs. So let's start with imagining a cat. Imagine you wanted to make that cat stop scratching, and to accomplish that you took away its scratching post. What would happen? Well, I'm sure that all the cat owners here know that that would not stop a cat from scratching. It would simply find something else to scratch. The carpet, the furniture, the precious leather armchair, or even you. So this is not the cat being malicious or destructive. The cat is simply doing what it is wired to do, no matter if it has a scratching post or not. Scratching would therefore be considered a behavioral need of a cat, since it is a behavior for which cats are motivated to do, no matter the context. So that at its essence is what a behavioral need is. So there are multiple definitions and philosophies related to this concept, but generally it is considered to be a specific behavior pattern that animals are motivated to perform, no matter the environment or even if the physiological needs that the behavior serves are fulfilled. And the fulfillment of behavioral needs is an integral part of animal welfare. When taking a look at different definitions of welfare, we see that it comes up in a lot of them, just with different names or different terms used. It's generally recognized that the freedom of satisfying behavioral needs is a big part of good animal welfare, and that if they're not fulfilled, it will have adverse effects. However, the term is not often defined or properly understood, which was a big motivation for me to do this review. So now that is covered, let's talk specifically about cows, as they're the main focus of my thesis and this paper. So their behavioral repertoire falls under 6 big categories. So no matter the context and where they live, they will always try to do grooming behaviors, resting, feeding, locomotion, socializing, and exploration behaviors. And the other part of my big question has to do with common housing systems. So before going into any of this, I'd like to say quickly that I am from Canada. So the types of systems, or more specifically the common systems, might be slightly different depending on your area of the world. So here in Canada, we have 4 main types of housing systems. We have cubicle systems that include tie-stall barns and freestall barns. So tie-stall barns have cows tethered to their stalls or cubicles by a chain, and freestall barns still include stalls, but cows are not tethered. We then have bedded pack barns or stall-free barns, where there are no stalls and cows are free to lie down wherever and however they want in the bedded pack. Thirdly, we have outdoor access, where cows that are usually housed indoors have access to an outdoor area, either at all times or during specific outings. And finally, there are some dairy cows that are kept on pasture. As for the link between behavioral needs and housing systems, well, as you might expect, the behavioral needs naturally fulfilled by the base environment varies quite a lot between housing systems. For example, if we compare cows kept in a tie-stall barn and cows on pasture, well, the cows on pasture have more opportunity for locomotion, for exploration, such as grazing and oral manipulation, and social behaviors, among others, compared to the tie-stall cows. So not every housing system is equipped to satisfy all of the behavioral needs of cattle. So putting all of this information together, what I was interested in observing was how the behavioral needs of dairy cows was studied in literature across all of these common intensive housing systems. And I had three main objectives with this. The first objective was to identify if papers measuring behaviors in dairy cows consider them as needs and which ones. The second objective was to see the what, where, and how of the study of behavioral needs of dairy cows. So we're looking at the behaviors studied, the housing systems used, and the methods employed to measure these behaviors. And finally, we wanted to see how the study of behavioral needs in dairy cows has changed over time. As for my methods, a scoping literature review was conducted following the PRISMA guidelines. I constructed my search strings based on the PCC framework, with the population being healthy dairy cows, the concept being behaviors, and the context being common intensive housing systems. So the scope was restricted in this way in order to establish a baseline of what was studied in this population. This led to the construction of this very long string in which we see all three concepts. So as we can see, we wanted papers that studied dairy cows in one of our predetermined housing systems, and we wanted them to study at least one behavior, which we can see a long list here. As a distinction of which behaviors are and are not needs depends on the author and the philosophy they follow, we chose to look at papers that measured behaviors that could be considered needs, which in this case is basically all of the behaviors in their repertoire. So that string was then put into Web of Science and Scopus and identified a grand total of 17,236 papers. This number was then thankfully reduced to 11,512 after deduplication in Zotero. From there, we did a screening of title and abstract on RAIAN. So this was done by two screeners in order to limit bias and make this process as objective as possible. The screening was based on my inclusion and exclusion criteria, and reasons for exclusion were attributed to each article. So these included language, type of paper, population, measure taken, field of study, and housing system. All conflicts between us were resolved by a third screener that was involved in the conceptualization of this review. And the final screening of the full text was done by me on Zotero to give us a final total of 144 articles included in my review. Data extraction and analysis using descriptive stats were done afterwards on Excel. So now that's out of the way, let's jump into my main results. So the first question I asked was if papers acknowledged the existence of behavioral needs and which words were used. This means I was specifically looking at the introduction to see if they used the word behavioral need or a synonym found in the green box, such as natural behavior, inelastic demand, species-specific behavior, etc. So we found that only 15% of my papers measuring behaviors used one of the words in their introduction. I also observed that there were quite a lot of words used to describe behavioral needs, such as high priority behavior, normal behavior, important behavior, etc. And these were found in 13% of my papers. But the big finding here is that the great majority, with 72% of papers, did not mention the existence of behavioral needs or the fact that the expression of these behaviors are important for the animal. So this shows us that there's a lack of common terminology, as there are 13 words here used to represent the same thing, and that there's still a lack of behavioral papers that discuss the importance of these behaviors. As for the second objective, let's start with the what. So which behaviors were studied in dairy cows? So in this weighted graph here, the number of papers measuring behaviors in each of the categories is shown and represented by the size of the bubble. So we can see that resting, feeding, and locomotion are by far the most measured. This could be due to the fact that they are easily observable behaviors that tend to occupy the time budget of cattle the most. As for the lines in the figure, they represent the number of papers that measured combinations of behaviors. So we can see that resting, feeding, and social behaviors were often measured together. This could be because they are very connected, as lying stalls in the feeding bunk have been shown to be the most common areas for competition, which would fit into social behaviors. Finally, grooming and exploration have been the least studied categories, and they were never studied alone. This could be due to the fact that housing systems are not inherently designed to fulfill behavioral needs, and the base environment of the cows does not provide ample opportunities to engage in grooming or exploration, which then makes them hard to observe in the home pen. There are also not behaviors that can be measured with technology, which I will discuss later in this presentation. Now let's jump into the where, so in which housing systems these behaviors were measured. In the bar graph here, on the x-axis, we have the different types of housing systems. So we have cubicle systems, bedded pack systems, outdoor access systems, and pasture systems. On the y-axis, we have the number of articles. As for the colors, the blue bands represent the papers that only used that one housing system, and orange represent papers that compared multiple housing systems. So we can see that cubicle housing was used the most, which is unsurprising as they are the most common. What did surprise me though was that outdoor access systems were used more often than bedded pack and pasture. This could be because of legislation changes around the world, such as in Canada, banning the continuous tethering of cows in tie stalls, pushing for alternatives such as outdoor access, which is an easier and more cost-effective way than changing barns completely. Finally, there were a lot of papers comparing bedded pack housing to cubicle housing. This could show the emergence of this system as an alternative to cubicle housing. So continuing with the second objective, we are now at the how. So what were the methods used to measure these behaviors? There were two main methods used in my papers. We had manual annotations and sensor-based technologies. Manual annotations were the most popular and included live observations, where an observer directly observes cows, either with continuous observation or with scan sampling at intervals. Some papers also used video annotations, where the observer annotates the behaviors of cows from video footage, sometimes with the help of a software such as the Observer or Boris. As for sensor-based technologies, there were three main types that I observed. We have activity monitors, which were the most popular. These were either attached to the leg or the collar of the animals, and they could automatically detect lying, standing, and sometimes a number of steps. The second type was eating and rumination recorders, which were either on the collar or on the jaw strap like in the photo, and these could detect jaw movements and head positions in order to infer if the cow is eating, ruminating, or resting. Finally, the least used method was automated feed bins that can detect feed intake and duration and frequency of eating bouts for each individual cow. To dig a bit deeper into the methods used, I wanted to see how the use of manual annotations, which is shown in orange in the graph here, and sensor-based technologies, which is shown in blue, change over the years. So on the x-axis we have the year published, and on the y-axis we have the percentage of articles in my study that used either. So we can see that the first study to use technology was published in 2002, and they used what they called a telemetric localization system. After that point, the use of both technology and annotations fluctuated between 33% and 100% of the papers published each year, but never going down to zero. After 2002, the average percentage of papers that used manual annotations was 65%, and for sensor-based technology was at 62%. So we can see that sensor-based technology was rapidly adopted, but it never replaced the use of manual annotations. In fact, there were multiple papers that used both at the same time to measure different behaviors. But finally, for my final objective here, I constructed this timeline that demonstrates how the study of the behaviors of cows has changed in time based on the papers included in my review. So historically, dairy cow research has been mainly focused on production and health. Housing choices were based on milk production, and behaviors such as grazing were measured to explain increases or decreases in milk production. Therefore, when it came to the study of dairy cow behavior, it was very much focused on the behaviors associated with production and health, namely feeding and resting behaviors. In the early 2000s, there was a rise in the validation of sensor-based technologies in cows that could automatically detect step counts and lying bouts, which led to a rise in the study of locomotion in cows, as it suddenly became much easier as it was done automatically. Additionally, around that time, there was an increase in the general public's interest in animal welfare, which was demonstrated in questionnaires, where people put importance on cows having access to the outdoors. And this could also explain the increase in locomotion studies, as the direct effect of an increase in freedom of movement would be an increase in locomotion. And later, in the 2010s, when the concepts of animal welfare and behavioral needs started being applied to farm animals, there was more interest in the addition of environmental enrichment to improve welfare. For example, the addition of brushes, which led to the study of grooming and exploration. And finally, for the path forward, I've observed an emergence of studies using AI models to automatically detect behaviors. So there's still work to be done to validate those models, but it is looking quite promising. So with all of this information that I've presented so far, this literature review established a base on behaviors that were studied in healthy dairy cows across all housing systems. It highlighted that the study of behaviors of dairy cows has come a long way in the last decades, as researchers have gone from measuring only health and production-related behaviors to now including the welfare-related ones. It also showed how quickly the adoption of different methods was done and is promising for the future. However, there is still work to be done when it comes to the definitions and the terms used to define behavioral needs in the literature, since there's still confusion and a lack of consistency that is observed in this review. There's also a lack of papers measuring behaviors that occupy the time budget the least, such as exploration, grooming, and social behaviors. But all in all, my results seem to indicate that we are moving in the right direction to support future guidelines, allowing implementation of improved practices in different fields of application, and in this case, the care and housing of dairy cattle. I would quickly like to thank my supervisor, my committee, and my lab members for their guidance, Lilou Vautrin for being my second screener and going through 11,512 articles for me. Thank you so much. And of course, my funding sources that make my master's possible. And of course, thank you for listening to my presentation. I'm looking forward to your questions.
Thank you for that talk, Catherine. Just give you a second to pop your camera and microphone on. Hello, nice to see you. So I'm going to jump in with a question or two first, just around the behaviours included in your search for your study. So you had, when you showed us your search terms, you had quite a lot of behaviours identified there, and I was curious what approach you took to deciding which behaviours to incorporate into the search.
Well, the main thing that guided us here is that because there's so many definitions and philosophies regarding what's considered a behavioral need, what's not considered a behavioral need, we kind of just wanted to include all the behaviors that could be considered a behavioral need in cattle, depending on like pretty much any philosophy that you follow. So it did end up being a lot of the behaviors in their repertoire. So most of them are in there, just to get a broader view there and not end up ignoring some that are important.
And I guess tying into that, so you highlighted the key behaviours that were observed. Was that all of the behaviours that were observed, or were there perhaps some that were just reported on very low frequencies? The individual behaviours, so we had the chart that showed us the feeding and the resting behaviour being really common. Were there any behaviours from that big list of individual behaviours that perhaps were reported but just not very commonly?
If I'm understanding it correctly, there was like exploration behaviors were not often measured. I think they came up in 9 studies. So either that was sniffing the ground, or in Tysol barns, it could be like sniffing the railings and stuff, but those didn't come up very often. It was mainly the resting and the feeding behaviors that came up.
Thank you. So I've got a couple of questions on here as well. So Baz has asked, did you have any explanation for the need for researchers to combine monitoring with sensors and with direct observations? Is this mainly linked to perhaps social interactions, and would automatic monitoring proximity to other individuals potentially be a solution for that?
That's a really good question. Yeah. So I did see them being used together, the manual annotations and the technology together, mainly in order to be able to measure a bigger, like more behaviors. So technology has been really used to detect lying, resting, number of steps, and eating, but for social behaviors, are really not that great. So there has been more studies using AI models to detect, proximity, like you mentioned. So you could detect like which animals spend more time together or more closely, but it is very difficult in that way to determine if they're doing affiliative or agonistic behaviors. So you can tell that, okay, animal A and B do spend more time together, but you can't tell if it's if they're fighting or if they're allo-grooming and stuff like that. So I do think we're not quite at this part in technology where we can completely disregard manual annotations, because they do give us a lot more information. But yeah, technology is evolving really quickly, so it should, in the next few years, there'll be different things there.
Thank you. As we've just got time for one more quick question. So Berta's added a question to the chat. So has the welfare of dairy cows in practice improved significantly in the last decades, or has increased production yield resulted in the findings standing still?
I do think if we look at legislation, there has been a bit of changes. I can't really talk for the world, but in Canada, there's been a lot of work done to try to increase the amount of freedom of movement given to cattle. So there's been a big change that as of 2026, so next year, the cattle, it'll be prohibited to keep them tethered at all times. So we are trying to really move away from tie-saw cows and give them more access to exercise and movement. It is slow, but hopefully in the next few years there'll be more.
Fantastic. Thank you so much, Catherine. Really appreciate your time there. And we'll move on to the next talk. Thank you. So our final speaker for session 2 is Richard Bennett. Richard is talking to us today on a talk about a method for assessment of the economic valuation of improvements to farm animal welfare for use in policy appraisal. So we'll hear from Richard now. Thank you.
I'd like to present to you a method for the assessment and economic valuation of the benefits of improvements to farm animal welfare for use in policy appraisal. Why is this research needed? Well, the Better Regulation Framework and the Treasury's Green Book sets out the requirements to assess the impact of any new policy or change to policy. And a cost-benefit approach is needed. In the past, it's proved difficult to monetize the benefits of improvements to animal welfare. And so it's been very difficult for policymakers to show the value for money of new policies to improve the welfare of animals. There's currently no universally accepted method on how to monetize animal welfare benefits. Previously, benefit calculations, if they were done at all, required separate and costly consumer willingness to pay surveys, which were not transferable across policies and usually weren't comparable with each other because they used different methods and valued very different things. Essentially, our research had two main work streams to it. The first was animal welfare assessment, and this involved the development and use of a structured expert elicitation welfare assessment protocol to provide welfare scores on a 0 to 100 scale for a series of different policy scenarios for 6 different farm animal types. They were beef cattle, broilers, dairy cattle, laying hens, pigs and sheep. Parallel to this was the economic valuation work stream. This involved estimation of households' willingness to pay for increases to the welfare score using a choice experiment survey method applied to a representative UK sample of over 3,000 households. So the link between these two work streams was the welfare score. The welfare assessment protocol involved an expert panel of 13 independent welfare experts. The protocol itself was based on a combination of the 5 domains model of welfare and welfare quality. So the panel looked at 12 welfare criteria, similar in description to those used for welfare quality, which were then aggregated into 4 different welfare principles: good feeding, good health, good housing and appropriate behaviour. and then they were aggregated again into a single welfare score. The welfare scoring system used by the panel was on this 0 to 100 scale, where 0 denotes extreme suffering of the animals involved, and 100 would denote the best possible achievable welfare. And the idea is that the panel would score the welfare status of animals before a policy change, and then score what they think the welfare status of animals would be after a specified policy scenario was put in place. Really important considerations for our protocol was that it took an animal-centric approach, and it's valenced, so it considers the likely positive and negative experiences of animals throughout their lives. So there's a focus on what's important to the animals, or at least what we think is important to the animals. It considers the affective and emotional states of animals for each of the 12 criteria, and it considers the opportunities that animals have for positive welfare experiences and not only for the avoidance of negative experiences. And as I said, it involves the whole life experiences of animals from birth to slaughter. The method used to elicit the expert panel's scores was based on the IDEA protocol, which has been used widely in environmental and other studies. The idea is that the panel shares information amongst each other and the facilitators share information with the panel. The panel then independently provide first round welfare scores for the scenarios presented to them. These are anonymous scores, and the panel is not allowed to discuss their scores at this stage. Then they're informed about each other's scores, again anonymously, and they meet to discuss those scores. They're then given the opportunity to provide a second round of scores, and these scores then become the final scores, which are aggregated to produce weighted scores for principles and the single aggregated score. The panel then reviews those scores to make sure that they are happy with them. Here is an example of the panel's scoring related to colony cage egg production. You can see the welfare criteria on the left and the panel's mean scores for each of those criteria on the right-hand side. I'm not going to go through each of these criteria scores, but I would like to draw your attention to the principal scores, and particularly the values in bold. These are the weighted values for each of the 4 principles. And you'll see that the arithmetic means are shown in brackets below those, and you can see that the Showcase aggregated values are lower than the mean scores. This is because they are weighted using a Showcase integral aggregation method. The weightings were elicited in a separate exercise from the panel. But what is worth noting is that the Showcase weighting gives greater weight to low welfare scores compared to high welfare scores, on the assumption that you can't compensate for low welfare scores by high welfare scores in another area. Here is a slide showing a comparison between the colony cage egg production scenario that you just saw the scores of and one for free-range egg production. Perhaps as expected, you can see that the score for free-range egg production, the single score shown on the right-hand side, is substantially higher than that for colony cage production, and indeed all of the parameters, actually apart from health, are substantially higher for free-range egg production than they are for colony cage production. Let's turn now to the economic valuation part of the method. We used a stated preference choice experiment survey. The design of this was informed by 4 exploratory surveys, 6 focus groups and 8 verbal protocol interviews. We wanted to make sure that we got the design of the survey and the questionnaire right. And we use people's responses from the survey to estimate their willingness to pay for an increase in the welfare score of each of the 6 farm animal types as an increase in their weekly household food expenditure. And we surveyed over 3,000 household main food shoppers. The shoppers were given information on the welfare scoring system and asked for their preferred choice from 3 options in 12 different choice sets. I'll show you an example of a choice set in a moment. The survey also collected the usual sort of information of such surveys on the socio-economic characteristics of the sample, their age, income and so on, their expenditure on different animal products, their attitudes and beliefs in relation to animals and animal welfare, and questions in relation to their choice question responses. Here's an example choice set. Perhaps you'd like to think about which one of these choices you would choose. So people are asked to please choose one from the 3 choices shown below. Numbers represent welfare scores of the animals and products shown and the impact on your weekly food bill on the left. Welfare scores shown in red are the only ones that change in relation to current welfare scores in each choice. The first choice was always the status quo, the current situation. And so people can choose choice 1, choice 2, choice 3. Which one would you choose? Some findings from our survey. 86% of people liked the idea of regulation to improve the welfare of farm animals, government regulation. 80% wanted to see such a welfare scoring system on labels in food stores. 89% of people understood the information presented to them in the questionnaire. 83% were confident in their responses to the choice questions, and 74% trusted the scoring method described to them. The vast majority of those not in that 74% category wanted more information and were neutral. They neither trusted nor distrusted the scoring system. Here are some results in terms of estimation of people's willingness to pay. We used a hierarchical Bayesian logit approach which allows for diminishing marginal willingness to pay, and I'll explain that in a moment. People's willingness to pay here are in terms of pounds per household per year. So if we look at the first line of the table for laying hens, you can see the status quo welfare score was presented to people as being 50 out of 100. Their willingness to pay to improve the welfare of laying hens from 50 to 51 is £4.44 per household per year. If you look though at higher levels of welfare and people's willingness to pay, by the time the welfare score of laying hens is 80, that's the final column, status quo plus 30, so that's 50 plus 30 is 80. To go from 80 to 81 in terms of welfare score, people are only prepared to pay around £2 per household per year. And what that enabled us to do, I don't expect you to be able to read these, but what that enabled us to do is to produce these valuation tables that policy analysts can use to value any increase in the welfare score of animals brought about due to policy. So you can just apply these values and immediately calculate the benefits if you have the welfare score information. Here is an example benefit calculation. It relates to the transition of laying hens in colony cages to free range. This was assessed by the panel as being a welfare score change from 32 to 51. The willingness to pay associated with that was £17 per household per year, which multiplied by the number of households in the UK gives an annual benefit of just under £500 million a year, which equates to 20 pence per egg, which is well within the retail price range found in food stores. In conclusion, measuring people's or indeed animals' preferences is not easy. People don't express their preferences perfectly either in surveys or in actual markets. The economic valuation that we've undertaken is based on human preferences for animal welfare in relation to food production and not directly animal preferences. However, I think we have a practical, flexible and relatively rapid appraisal method. It's animal-centric in that it considers what is thought important to animals based on animal welfare science. In a sense, it estimates pure animal welfare willingness to pay values, not linked to specific production practices or particular contexts. The willingness to pay values that we have are transferable across policies, and benefit calculations are comparable, unlike previous studies. I really hope that this explicit quantification of the benefits of policies to improve the welfare of animals should help strengthen the case for such policies. Thank you very much. Any questions?
Fantastic. Thank you very much for that talk, Richard. Let's give you a second to get your camera and microphone on. Hi there, Richard. So we've got quite a few questions. We've got quite a few questions have come in and just a few minutes for them. So I'll pick a few out and then hopefully the rest can be addressed possibly elsewhere or by contacting you. So there were a couple of questions that came in early on around the methods that I'll just start with first. So how did you make sure that the cumulative animal welfare across the lifetime was calculated in the same manner by all participants? So I think that relates to the specialist stakeholders part of the study.
Yeah, that's a good question. What we've got here, of course, is the panel is asked to look at the whole population of animals in that particular system, let's say colony cage production across the UK. So they have to take into account numerous different factors which you can't easily do in a sort of easily calculated way. So we rely very much on the expert panel to weigh up all the different types of system and how prevalent they are within the population. So what the panel would do, for example, one of the scenarios they did was relating to the use of farrowing crates in pig production. They would say, well, okay, throughout the life of the pig, the sow spends around 25% of her life in a farrowing crate. We take that into consideration, that it's 25% of the animal's life. In the case of another scenario we looked at was lamb castration, then that's a smaller amount of time of the lamb's life, actually affected by that process, and so there's a less of an impact on the welfare score. So, you know, it's not easy what the panel had to do, and they did a fantastic job. I actually was concerned about whether this protocol was going to work, and it developed over time with the panel. The panel helped develop it, and actually it worked incredibly well, better than I actually thought it might.
Thank you for that. I'm going to continue with just one more question from Hugh around the scoring again by those specialist stakeholders. So we were able to take account of the variability in scores that they then awarded to those different welfare criteria. For example, was there any strong agreement between the raters or not?
Yeah, another good question. It varied. Now, what we didn't have was stakeholders as such. We very much had an independent panel of experts who had no prior expectations of what these scores should be or what they wanted them to be. So they came to them as independent experts looking at it objectively. Yeah, there was quite a variability of scores, and that's because we chose the panel expressly to come from different backgrounds. So some were practicing vets, others were academic welfare researchers, and so on. There was absolutely no requirement for the panel to reach consensus on scores. So we used that variation. And actually with a reasonable sized panel, and the recommendation is to have 12 or more, that variation is a strength and not a weakness. So they didn't always agree on particular scores, but that's often because during the discussion, you know, they talked about having quite different perspectives sometimes, and I think the panel really enjoyed learning other people's perspectives on particular welfare issues.
Fantastic. Thank you so much, Richard. There are a lot more questions for you, Richard, but unfortunately we're out of time. So we're about to close this session and move on to the break. But thank you again for your talk and for all the others who did a talk in this session. Hopefully some of those questions can be posted more centrally for addressing, or people can reach out to the speakers as well to find out more about those answers. So thank you very much.